The replacement of failing organs with lab-grown fully functional human organs is a challenging goal that science has yet to achieve, but recently significant advancements have been made that may one day make this possible. The biggest issue for organ transplantation is organ rejection. This is when the immune system of the recipient recognizes the new organ as foreign and starts to kill the cells like it would during an infection. To prevent organ rejection, transplant patients need to take drugs to suppress their immune system, which leaves them vulnerable to infections and cancers. Even with these drugs, the average life of a transplanted organ is 12 years, so some patients will need multiple transplants in their lifetime. But if scientists could somehow grow a new organ from the patient's cells, there would be no need for immune-suppressing drugs, and the organ should last them a lifetime. Scientists are working towards this goal by studying how organs develop. In humans, organs develop from pluripotent stem cells in the embryo, called embryonic stem cells. These cells have the potential to differentiate into any cell type in the human body. As the embryo grows into a fetus, these cells multiply, differentiate, and organize themselves into mature organs. This process can be mimicked in the lab by growing embryonic stem cells with specific growth factors to coax these cells into organ-like structures, called organoids, or mini-organs in a dish. This is done with a 3D culture system where the cells can grow in 3 dimensions within a gel of extracellular matrix, like the one found outside cells in the body. Due to ethical issues with the use of human embryos, this research is tightly regulated and funding is restricted internationally. This led researchers to try to find alternatives. It was known that some organs like the intestines can regenerate their whole lining in a matter of weeks, and it had long been accepted that adult stem cells in these organs were the source of this ability. Once the location of these stem cells was identified, they were isolated and grown with specific growth factors known to be involved in intestinal development, and it was discovered that these adult stem cells residing in the organs could also develop into organoids. These organoids have the same cell types and structure as the lining of the intestine. These adult stem cells are already committed to becoming intestinal lining, so they will only make intestinal organoids. Around the same time, scientists had discovered that they could create pluripotent stem cells by reprogramming adult cells with specific growth factors. Instead of needing a biopsy of a specific organ, a fibroblast from a skin biopsy can be used to make organoids of any organ type, including intestines, stomach, kidney, heart, and brain. This also overcomes the ethical issues of using human embryonic stem cells, as these induced stem cells can be used instead. Organoids are also being used to advance many other areas of science, such as drug testing, cancer research, infectious disease, developmental biology, and regenerative medicine. Organoids give researchers access to human organs that are otherwise inaccessible, and they also more closely reflect human biology than animal models.